Wow. Good evening, everyone. A little loud, sorry. Good to have you. Welcome. This is our final Science Cafe of the summer season. So we will transition to having one Science Cafe a month starting in September. So it's been fun to have a busy, packed summer, but we're also ready for a little breather. So thank you all for being such good supporters of our Science Cafe. And tonight we are very excited to have a conversation with Dr. Yannic Becker, who is going to talk to us about the importance of maintaining a healthy vascular system. And he's doing some very interesting research that is really shining a new light on what that process looks like. And so he's going to tell us a little bit about that a bit later on. But before we get to that, we want to talk to you just a little bit about some fun facts about the vascular system. So I was saying to Yannic that I used ChatGPT before I came and said, okay, tell me some fun facts about the vascular system that I can share with the audience. And the number one fun fact, tell me how many miles do you think the average human vascular system is, the whole thing?
Can you hear it?
Mm-hmm. And those of you online, feel free to chime in, raise your hands, let us know. Any guesses? You got to make a guess. I can see you're thinking. Okay, 200 miles. Who thinks?
The moon and back. Somewhere in between.
He's absolutely right. The average, this blew me away. The average is 60,000 miles. Like, how is that possible? And I asked him, I was like, is that true? And it is. So when you think about all those teeny tiny little capillaries, you know, I was reading that even some capillaries are so tiny that the red blood cells have to actually turn into a different shape in order to get through those capillaries. So fascinating. And we're going to learn a lot more, but I wanted to just kind of share that with you because I thought it was really, really interesting. For those of you who are, say, non-scientists, when we talk about vascular disease, what comes to mind for you? What do you typically think about when you think about vascular disease?
Heart attacks.
Heart attacks, right. Yeah, exactly. Heart attacks, aneurysms. What else?
Strokes.
Strokes, exactly, yeah. But what I didn't always think about were diseases like diabetes, right? I didn't necessarily make that connection between diabetes and a deterioration of the vascular system, but yet we know that that is a huge issue and can lead to vascular disease itself, you know, an increase in heart attacks and strokes, certainly an increase in kidney damage and disease. And so that's a big part of what Yannic is interested in, he and his group. And before we get started, I want to ask Yannic to tell us just a little bit about his background. So we're very proud of him because he's a newly minted PhD. He received his PhD through our graduate program and the partnership with Hanover Medical School, or I shouldn't say Hanover Medical School, but the Hanover University there, and in June received his PhD, and now he's back with us as a postdoctoral fellow. So congratulations.
Thank you. Thank you.
Great to have you. So tell us a little bit about what the Haller Lab, you work with Dr. Herman Haller, who is here at MDI BL. Tell me a bit about what your group is focused on.
Right. I think you already mentioned it a bit. So we are interested in the cardiovascular system, and probably what you see all the times in cartoons or graphics is something like this. You see this major arteries in red and veins in blue, and this isn't really the truth. This is very superficial. I mean, these are the big blood vessels that transport the blood into the tissues where it is actually needed. And Jerry already alluded a bit to that by saying that the real action of the vascular system is taking place in the capillaries. And I think this middle picture is quite fascinating.
Wow.
This is from an exhibition called Body Worlds, and actually it's also here in the US. I've seen it in Germany, but those of you who are interested, you can see these exhibits I think at the moment in Philadelphia and in Houston. And so it's the whole human and in general mammalian anatomy, and they're quite fascinating exhibits there. And also those of the vascular tree. So this is the complete vascular system here. And you can see there is much more when you compare the left picture to the right one here. There are so many tiny blood vessels, and these are the capillaries. And they make up these 66,000 miles, or I like to express it in square meters. So 1,000 square meters is actually the surface that it's estimated. We don't know exactly, but it's roughly a quarter of a soccer field, just to like make it a bit more visible.
Wow.
So it's a huge area that we are dealing with. And that is actually what the Halle lab is interested in. And here I have a tiny, this is a cartoon of how one of these tiny capillaries actually looks like. And it's made of a cell type called the endothelial cell. And the endothelial cell is the cell that forms the blood vessel. And this is the primary cell type that we are interested in and working on.
That's great. And so the reason I like to do these is because I always learn 10 times more than I knew before. And so when I think about blood vessels, that's really the image that pops to mind, right? This is what we all learned in our textbooks. But in reality, the surface looks a little bit different, right?
Yes.
Of the interior. And you have some fascinating pictures that we're going to get to. But tell us just a little bit more, Yannic, about the endothelial cells and kind of your interest in something known as the glycocalyx, which is the coating, right, of those blood vessels.
So maybe the first question is, what do we need a vascular system for? And I think we should maybe start with this first. So any of you, I mean, some major functions are known, I believe, but what do you know is, why do we need the vascular system, or why is it important?
Bring oxygen.
Yes, that's the major one that comes to mind first. That's why this is the red blood cells, they transport the oxygen from our lungs, where it's getting oxygenated, into the periphery, and there the red blood cells release the oxygen, and so the cells around it can actually like breathe or work. Yes. Nutrients, exactly. Yeah. So besides oxygen, our cells need also sugars, amino acids, fatty acids. All these components are transported via the vascular system. Exactly. Yeah. Great.
Get rid of waste material and CO2.
Yes. So this is the other thing that's often forgotten, that it's not just to transport oxygen to our cells, but also to get rid of the carbon dioxide and get it back to the lung and where we breathe it out. This is another one. Yeah. And then also something like hormones. So communication between different tissues. One example is, for example, you eat a couple of crackers out there and you secrete, your blood sugar rises and you secrete insulin. Your pancreas secretes insulin. And this insulin is actually acting in other tissues, primarily the skeletal muscle and the liver. And then these tissues, they respond and they take up the sugar that is in the bloodstream.
Fascinating.
So
far more complex than just a pipeline.
Oh, definitely. Yes.
Fascinating.
Yeah. Sorry. And then one other thing is this is the transport function. And then Jerry, you already mentioned that there is some kind of a barrier around or in our blood vessels, which I will get to in a second. But the barrier function itself is very important for the vascular system too. So when we think about, for example, we have also an immune system, which is continuously circulating in our vascular system. So when you go to the doctor, you also get white blood cell counts and not just red blood cells. So the white blood cells are the immune cells. And this system is also tightly regulated, not to like go into each tissue where it wants to, but primarily the immune cells are supposed to stay in the vascular system and only exit the system when you really want them to exit, like when you have an infection. So there is a barrier in the vascular system forms, and this is quite important too.
It's fascinating because we don't often think of the vascular system as being permeable, right? I mean, we tend to just think of it as being kind of a pipeline.
Yeah.
But
Yeah.
in reality, it's constantly allowing things to cross that
Yeah.
that membrane.
Yeah.
Fascinating.
Yeah.
Very cool. So tell us a little bit more about, oh, sorry, question?
It's important in temperature regulation. It opens to cool us in the summer and it closes peripherally to keep us warm and in the wintertime.
Yeah. We kind of skipped over that, but the transport of heat is very important. Like when we exercise, we generate a lot of heat in our muscles, and this heat is getting transported via the circulatory system to the periphery, and there we can then, the heat gets out in the form of sweat eventually.
Nervous system controls blood flow, correct?
Yes, that's correct. Yeah, yeah. So it can like make vessels dilate or contract. Exactly.
So tell us a little bit more about this glycocalyx that we've heard about that you and your lab are interested in studying.
So I can show you.
There you go.
Oh, it is responding. Okay. So this you've seen before, this top left image of how one of these capillaries looks. We have the endothelial cells and we have a lumen, and the lumen is where the blood flows through. But the reality is when you look through an electron microscope, in this case, is you see the architecture is the same. You have the lumen here, but you have this brush-like structure, which is enlarged here in the middle part, that is covering our blood vessels. And this structure is called the endothelial glycocalyx. And I gave the slide the name, the blood vessels are sugar-coated, because actually the macromolecule that forms this glycocalyx are sugars. And I think this is something nobody really is aware of. Not even scientists are aware that this structure exists. Like many people in the field, even of vascular biology, kind of ignore this. But we believe that this structure is important, and now we have also collected some evidence over the last year that it really is for vascular function.
And Dr. Haller was saying that there is this field of sugar biology, just trying to understand these different elements, which is fascinating, a whole area that we don't really know yet a lot about. So that's great.
Yes, all mammals. We come to this later, but we work with zebrafish, and they are vertebrates. They're not mammals, but they have this structure. And you can look into other animal like, for example, Drosophila. They also have these structures like fruit flies or worms. They don't even have a vascular system, but their cells are covered by these structures too.
Discuss why and what they do.
Yes. Yes. So a question for you, does any one of you know heparin? Or has any ever used heparin, or a doctor prescribed heparin to you, the drug?
It's an anticoagulant? Is that what it does?
Yes, exactly. I've seen Yeah, and heparin, how do you apply it? Can you take it like orally, or is it? It's injections, yes. And the reason why it's injections is because it's also a sugar. Heparin is a sugar, and if we would ingest it, our gastrointestinal tract would degrade it and would make use of it as a source of energy. But the drug itself, the heparin, would never get into our blood vessels where we actually need it. And you said it's an anticoagulant, and that's exactly right. And this structure is a heparin-like molecule that we are dealing with. So I thought maybe some of you have taken or heard about heparin as an anticoagulant, and so you can imagine that we have this structure, heparin, naturally in our body, and actually it is all over the place in our blood vessels. And there it has the same function. So it's an anticoagulant because we don't want our blood to coagulate. It's dangerous, as you said, heart attack or stroke. This is like the terminal outcome of this if this is not working properly. So we have to control this system very tightly and keep the blood in an anticoagulant state. And only if we have an injury somewhere, then we want the blood to coagulate to close the blood vessels.
Anticoagulant these days seems to be Eliquis.
Eliquis, yes.
And is that function the same way?
It also acts in the coagulation cascade. There are many, many proteins involved there. But as far as I know, is that Eliquis is inhibiting a protein directly of the coagulation cascade, which is a bit more, it happens a bit earlier in the coagulation cascade to where heparin is actually acting.
Mm.
Heparin is actually also binding to another protein. It's antithrombin 3. And this complex, this is a very specific now, but this inactivates thrombin and factor 10.
Mm.
Yeah, and Eliquis is directly acting on factor 10, I think.
And aspirin the same way, or is that a different?
Aspirin is cyclooxygenase inhibitor, and this happens. This is more important for the platelets of the blood. So you know we have also platelet. We have the coagulation system has a protein component and it has a cellular component, and the cellular component are the platelets in the blood. And aspirin is acting on cyclooxygenase. This is how the protein is called, and this is in platelets. So it's acting in another corner of the whole cascade. Yeah.
Awesome.
Yeah.
Somebody asked a question, could be repeated? Oh, no, people are having a hard time hearing. Okay, sure, you can do that.
So we were just chatting about some of the different blood thinners or anticoagulants that are on the market and how they work. And so tell us, we've talked about the fact that the glycocalyx sort of, or the endothelial layer acts as a barrier in terms of the permeability of the blood vessels. And you mentioned that there's still a lot that's unknown about exactly how this glycocalyx functions. Do we know, is it a filtering system? How does, we see all these amazing structures. Do we kind of know how it works?
So basically you see, like, you have this brush-like structure that is sitting on top of the cells. And when you think about molecules or cells that want to or have to interact with the cell itself, then this is quite a far way from the lumen to make it there. And this is what these structures are for. They form kind of an initial site for binding of circulating molecules or cells. Even cells can bind to these structures. And then this is like an initial tethering, and then more interactions are happening, and they slow down when the blood is flowing. They slow down on the surface, and then they make contact with the endothelial cells beneath. And that facilitates then, for example, that these cells can migrate out of the blood vessel into the tissue.
And so this is what a healthy blood vessel looks like.
Mm-hmm.
And so what happens in the presence of disease? When do things start to go awry?
So for this, actually, like, we have one more slide. Yeah. So here you can see again the same structure I've shown you before. And there were many studies done over the last, I would say, it really came to mind that the structure exists like 25 years ago, around the year 2000. And in different vascular diseases, what we see is that this layer disappears. It's not there anymore. The endothelial cells are not covered by this layer anymore. And researchers and scientists were wondering, of course, what does it mean? Like, is this a physiological response? Is this a good thing that this layer disappears? Or is it maybe a bad thing? And it was evident then that the more and more disease states were found, like atherosclerosis. You mentioned diabetes. Sepsis is another very acute vascular disease. That under all these circumstances, the glycocalyx disappears. And so this then, the idea came up that maybe this layer is good to have on our blood vessels. And maybe can be a strategy to kind of regrow the glycocalyx and protect it. Yeah.
And so that's really what your project has been about, right? Understanding exactly what's happening in this process and what are the mechanisms that exist to protect or preserve the glycocalyx.
Yeah, yeah.
Right?
So one thing we know about when the glycocalyx disappears is that the vascular system gets more permeable. And does any of you have an idea of in which tissue or organ this could be a particular problem? The brain. Why the brain?
Hemorrhaging in the brain. That's
Yeah,
not a good thing.
yeah. So our brain has naturally a very tight vascular system. You've heard about the blood-brain barrier. So the body really tightly controls what gets into the central nervous system and what has basically which molecules have access. So it was actually found, interestingly, that the glycocalyx there also plays major roles, but if it disappears, it's not that suddenly all the vessels are open and leaky and things can flow into the tissue. This was not the case, but it plays also important roles there. This is known. Yeah. Are there any other vascular beds that you can think of that are maybe not as tight as the brain or the blood-brain barrier?
Kidney.
The
Kidney. Yeah.
Pretty good guess.
Yeah. Why is the vasculature of the kidney not so tight? Like, why is it leaky in the first place?
It has to filter the
Yeah. So the primary function of the kidney is to filter our blood and get all the waste and all the products that we don't want in our body, get them out. So naturally, the vasculature has to be more leaky than, for example, in the brain. And what was found actually is that when we get this layer disappears in kidney vasculature, then the kidney gets way more permeable, and we lose a lot more substances that we don't want to lose normally when this layer is gone. And this then maybe you've heard about proteinuria or albuminuria. This is what it's called, is when protein ends up in our urine. And usually we want to keep the protein in our body. Same with the glucose. Usually we want these things in our, keep it because they're valuable for our body. Yeah.
Fascinating.
Skin.
The skin. I don't know if there were studies done of the glycocalyx in this skin vasculature, like or dermal vasculature. I think this was not really investigated so far. Like it's primarily really like organs, like kidney is often mentioned. Yeah. I don't know. The
Say it
liver.
again, Jim.
Yeah, the liver is another level of leakiness, I would say. So usually we can classify vessels in 3 categories, how leaky they are. We have the continuous blood vessels. These we have in the brain, like the blood-brain barrier is very tight. And then we have fenestrated blood vessels. This we have, for example, you mentioned in the kidney, where we have a certain degree of permeability, and then we have sinusoidal blood vessels, and they are extremely leaky, like anything, like even big proteins can like get in and out. And we have this, for example, in the liver, where you have a lot of exchange between the hepatocytes and the blood. Yep.
Fascinating. So tell us a little bit more, Yannic, about your particular interest. So we've talked about heparin, we've talked about the glycocalyx, but you're really interested in a very specific protein that kind of is involved in the process called heparanase 2.
Mhm.
Tell us a little bit about that.
So Heparanase 2,
Am I in your way?
yeah,
Maybe I'm
there it
blocking you.
is. So Heparanase 2 is a protein, and this protein is known to interact with heparin. This is where the name of the protein comes from. And so Heparanase 2 thereby then interacts with the glycocalyx, because the major component of the glycocalyx I mentioned is a heparin-like molecule. So we know that heparanase 2 is sitting in the glycocalyx. And this is actually what you see here. This is one image we took during our study, is you have a blood vessel here in green. This is a medium-sized blood vessel. And then in magenta, you have heparanase 2. And you can see that heparanase 2 sits on the luminal side of these blood vessels. And this is exactly where the glycocalyx is. So we know that it is there, this protein. And well, to be honest, when we started our research, we weren't really sure if it's really there. This is one of the things we found. And this was quite interesting. So we found a protein that is sitting in this layer, in this brush-like layer, and it was basically not characterized what it's doing there. And one idea was in the, sorry, was there a question?
No, I'm sorry.
Okay.
Sorry.
One idea was that heparanase 2 is a molecule that degrades the glycocalyx. It like leads to the state we've seen before, no glycocalyx on the endothelial cells. The reason why is because those of you who know, everything that ends with ASE protein is usually an enzyme. So something that catalyzes a reaction. And there is a heparanase 1 also, and heparanase 1 is known to degrade the glycocalyx. So it was a very valid assumption in the first place that, okay, heparanase 2 is just another molecule which degrades glycocalyx. And then over the years, it became clear that heparanase 2 can't do this. Heparanase 2 has no activity on these sugars. It can bind them, so we know it sits there, but it is not degrading them. And this is where this whole project started in the first place. So we have a protein that is sitting and interacting with these glycans, but we don't know what it is doing there. And we also did not know for sure that it was really in the vascular system. Yeah.
Cool. So tell us what it does.
So what the first discovery really was that it is there. And you mentioned before already that, or you asked, is it also in other species, this structure? And we found that we have heparanase 2 also in zebrafish, for example. It's also known to be in all mammals like mice and rats and all these, they have heparanase 2, but also lower vertebrates like zebrafish, they have a heparanase 2. And we did our study, and then we found, okay, heparanase 2 is really, it is circulating in the vascular system. And then our approach was to what happens if we take it out, if we delete the gene that encodes the heparanase 2, and do we see a phenotype then in the vascular system? And what we found actually is what we discussed before, is that the blood vessels become more leaky when heparanase 2 is missing. And that made sense to us, like also with the literature, that loss of glycocalyx in the first place could lead or increase vascular permeability. So this was then an interesting finding that fit with the literature. Of course, we didn't mechanistically know at this point how this works, like what leads to this leakiness, but at least this fit all together at this point. Yeah.
That's great. So tell us more about what you've learned about the role of heparanase 2 in terms of what can we do to
Maybe one more thing is we have a ton of different factors out there that want to interact with cells all the time, like certain molecules like growth factors, factors that would really open up our blood vessels and increase permeability. And we need to regulate this system. And so we came up with a hypothesis that maybe heparanase 2 is sitting there to regulate the access of molecules to the endothelium, and to sit there as kind of a shield, so to say, and say, we let you interact with these chains here, but not too much. You know, like we block the interaction at one point. If there is too much happening, like heparanase 2 is there and kind of shields it off. And this was our hypothesis of what, on a molecular level, this protein could actually do. And to test this, then what we did is we generated the protein in the laboratory. So we didn't look at it in a living animal at first, but we did a biochemical analysis where we purified the protein. And then we tested the idea of what happens if we have heparanase 2, and if we have these factors that induce the permeability phenotype. And does heparanase 2 prevent from the effect of these factors? And this was then indeed the case, that heparanase 2 is sitting there in the glycocalyx, and these factors that want to bind to the endothelial cell surface, they don't have access because heparanase 2 is blocking the binding sites there. And this was really then the major finding, and we showed this then also in zebrafish, that this is the way how it works in an animal with a fully functional circulatory system.
Yeah. So, you know, we know that, say, in diabetes patients, for example, often there is, you know, damage to the microvasculature or these small vessels. How does this finding maybe, you know, contribute to your understanding of that process, or possibly how to prevent it? Does it give you a different way of thinking about how we might maintain or preserve vessel health?
Yeah, so this is really like a new therapeutic avenue in this, because we know that this layer is good. I mean, this is the basic assumption, that if we have it, this is kind of a steady state our vasculature is in, and we want to maintain this state. So if we could use heparanase 2 then to kind of protect our blood vessels from these factors that act on it permanently to activate it, then that would be a new strategy to maintain vascular health. And you mentioned like the terms of prevention and therapy. So I think both strategies would work. It would be great to maintain our vasculature in the first place in this state. But of course, as a therapeutic, then when we have really the state of a degraded glycocalyx, highly permeable blood vessels, that we could apply the protein then, and could thereby then restore and rebuild a glycocalyx structure. I think I also have we skip this for the moment. This also. Yeah, so this is basically the idea that we have. We come from this state, and then we apply heparanase 2, which is now our model candidate, but maybe it's not ideal because it's a protein we deal with. It's a big protein. We don't know the half-life time if we apply it. How long is it actually circulating there? Can we modify it in some way to make it more stable? Are maybe shorter fragments of heparanase 2, so just the peptide, like for example, insulin is just the peptide. Is this a better option in terms of pharmacodynamics and pharmacokinetics to rebuild and restore the glycocalyx? And this is the end goal. So basically, yeah.
And you don't have any work to do to get to that point.
No, of course not.
That's great. Well, this is fascinating. Questions for Yannic from the audience? Yeah.
I'm wondering, is the glycocalyx the same all throughout the circulatory system? Because we have arteries, capillaries, veins, lymphatic vessels? And does it change with age?
It's very different from vessel type to vessel type. We know this already because the whole enzymatic machinery that builds these glycan chains in the cell is differentially expressed among different vascular beds. Yes. And we also have studies that show that during aging, this changes. One would assume that it goes down in aging, right? This would be but I think it is, we can't say this at this point because the structure it's so dynamic. Like this process of we build up the glycocalyx and we degrade it, this can happen within a couple of hours. And actually, when we have an acute phase, like an infection, we know that this layer rapidly degrades. It can disappear within an hour. We can do that in cell systems, and also it was done in whole animals. This is something that happens really fast. So it's very hard to then say this is really like an effect of aging, or was it just the circumstance at the moment? Yeah. Yeah.
Does Heparanase play a role in cancer? It's known that blood vessels of solid cancers, of solid tumors are quite different from normal blood vessels.
Yes, absolutely right. So
Repeat that question, sorry, so they can hear online.
the question was whether heparanase plays a role in cancer, and you're asking for specifically heparanase 2 or heparanase 1 and 2. Like, so interestingly, we see that, and this is where the heparanase 2 field started off. It came from the cancer field, and there is one group, they're working for a very long time in Israel on heparanase 1. And then they switched and also worked on heparanase 2, and they do all this work just in the cancer field. And what they see, and they've done numerous solid cancers by now, they always see the correlation that heparanase 2 goes down with the severity of the cancer stage. Yeah. Yeah. And heparanase 1 is a big player in the cancer field in general. So there are many attempts by now to inhibit the action of heparanase 1 as an anti-cancer therapy. The problem is that heparanase 1, same as heparanase 2, does so many different things, and that just inhibiting the enzymatic action of heparanase 1 seemed to be not an ideal concept of, or was not so well tolerated in patients. That was the major problem, like the side effects were too big by heparanase 1 inhibition in cancer. Yeah.
Is this type of therapy applied today, or in what avenue would it be?
So there is no drug approved by the FDA that targets specifically the glycocalyx. Yes. Yeah, even heparanase 2 could be used, but it's too early stage to, and this is why we think this is so exciting, because it's really a new therapeutic approach and something the pharmaceutical industry hasn't really picked up on yet to target the glycocalyx specifically. This has not been done so far. Yeah.
So given that, Yannic, what's next? I mean, you've got lots of questions, right? So where do you go from here? What's your next step?
So from the clinical, or like from the therapeutic perspective, one would really need a study, like still in animal models, but really to put the animals in a severe vascular condition, like induce an acute sepsis, for example, or induce diabetes, and then we apply the protein like the heparanase 2 itself, or we have peptides that are fragments of the heparanase 2, and we would see what's the outcome of this. And this would really be the direct experiment to validate that this is a therapeutic strategy, and then of course look at the side effects also. And this is actually why heparanase 2 is so interesting compared to a heparanase 1 inhibitor, because heparanase 2 is not directly acting on the heparanase 1. It is just blocking the action of heparanase 1. So it sits on the molecule that heparanase 1 wants to interact with, and this is the heparin. And it is not directly inhibiting the catalytic side. And we think that this is also one argument for heparanase 2 instead of a direct pharmacological inhibition with a small molecule of heparanase 1.
And so have you seen either in, mouse studies or where you've seen a reversal of a particular disease like diabetes? So if you can, encourage or preserve that, do you see a demonstrated improvement in a disorder?
We do. So we did a study before, and this was in sepsis. Sorry, I think there's one question. Maybe we can.
Just more on that, actually. I mean, do you see the direction of travel of this research going as far as preventing an aneurysm, reversing an aneurysm? Or is it more the type of cardiovascular disease like sepsis and diabetes?
I think it's prevention with this approach too, long term. It's really the goal is to keep our vascular system in a state where these things like these terminal outcomes like aneurysms or myocardial infarction, these don't even happen. We want to keep the vasculature in a steady, homeostatic, healthy state. And this would be the goal, yes. I think applying it when you have an aneurysm, it's probably too late by then, yeah.
How do we keep the system healthy?
How do we keep our vascular system healthy?
How do we keep it?
That's good. What are the tips? So managing our sugars would be one, right?
Yeah. Well, exercise and nutrition.
Yeah. As with everything.
Yeah. Yeah, but actually, maybe you've seen it in when you go to a grocery store. You see sometimes you have in the vitamins and supplement section, you have things like glucosamine. Maybe you read this, or chondroitin sulfate, like things like these. And these are actually building blocks of these sugars that form the glycocalyx. So they're already sold in the supermarket over the counter.
Interesting.
They are there. The question is whether they really end up in the vascular system in the glycocalyx. This is a whole different thing.
Right.
But in theory, it is known that these are beneficial. But we don't really know what happens if we ingest them. And so there are some over-the-counter drugs available that claim to be protective for the glycocalyx. So there is one company out there that was formed based on I believe it's a sugar mix of all these components that form the glycocalyx, and you can buy this, but it's not an FDA approved. So, and it's unknown how well it really it works when you apply it orally. So yeah.
Interesting. So how?
Blood test for heparanase 2 that you can have in your lab?
We don't. This is a great question because we don't have a reliable test at this point to measure the levels of heparanase 2. And the problem is that heparanase 2, I told you, interacts with this layer. So it's really hard to tell how much heparanase 2 we actually have, because when you take a blood sample, you only get what is flowing around. You don't get the proportion that is sitting in here. And we know that the proportion sitting in here is way bigger than what is freely flowing around. So it would be great to have a reliable test on this, how much heparanase 2 we have, how much heparanase 1 we have. But it's not used in the clinics yet. But what we do have, and I can go back for these steps, or what came up also like during the last years, is that there is one study, one group in the Netherlands, and they were also very interested in the glycocalyx. And they were wondering if they could measure the state of our glycocalyx. So how thick the glycocalyx is, because a thick glycocalyx we think is a good thing. And so they developed a tool. It's actually a tool where you put what you put under your tongue, because there you see you have the blood vessels are very close to the surface and you see them very well. So with a microscope, you can see here, this is what you see, all the tiny blood vessels. And you can also see the red blood cells flowing through there. And what they measure then is they measure the diameter of the capillary, and then they measure how much of the space there is occupied by the red blood cells. Assuming that the red blood cells cannot cover the space where the glycocalyx is, because the glycocalyx is blocking it. You can see the blood cells only flow in the middle. Here, this region, they don't get to. And then when we have a diseased glycocalyx, this column where the red blood cells can flow would get bigger. And this is called the perfused boundary region, PBR. So there are tools out there, and people try to make use of this to non-invasively look at the state of your glycocalyx at this moment. Or like, I don't know, maybe you're a smoker. I know that they've done it with smokers, and they see, okay, we have a decreased glycocalyx in smokers. But we also know that it changes a lot after eating a big meal. We also know that this can change the dynamics of the glycocalyx, because a lot is happening there, like all the time. And this makes it so challenging that this layer can rebuild and degrade so rapidly.
Awesome. Any other questions for Yannic?
This is great. What are you most excited about, Yannic, when you think about where this project goes next?
I think it's really that this approach was not, or that we have now a tool with Heparanase 2 to really regulate the glycocalyx directly. And we have something that our body makes naturally. We know that we have Heparanase 2, our cells can make it, but we don't really know why they make it, at which time point, like under which conditions do our cells make Heparanase 2, and how do they regulate the glycocalyx with this. And I think that's super exciting that we have this tool now in our hands, like with the protein itself, to play around with this. And we can choose the model whatever we like. We can work in zebrafish, we can work in mice, we can work with tissue cultures, or we can characterize it more on a very detailed level with like biochemical approaches. And this is great. And the other thing is that all these animals have a Heparanase 2. And we have now a fish, a zebrafish, where we deleted the Heparanase 2, and we see also other things that are happening. Not just the vasculature is affected, but we see that it has an effect in developmental processes. We also see that it is important for liver physiology and for iron metabolism. So the glycocalyx is, I talked about it in the sense of the vascular system, but all our cells, no matter which cell type it is, it has a glycocalyx. And so I think that's super exciting. And the field of glycobiology in general is far behind all the other fields of RNA biology or protein biology. And this is really a new branch of biology here that we now try to transfer from the dish and from a biochemical level, which was done the past 50 years, to really get it in living animals in vivo.
So, we all, of course, want to talk about a therapeutic, right? A drug that we can take that'll protect us from these things, or, some preventative steps that we can take. But I'm just curious, given the fact that there is so much that we yet really don't understand about how this process works, when we talk about the importance of basic research, which this really is, what would you say to whether it was a funder or, just someone in general about why investing in this kind of research is so important?
It is just a complete different avenue, I think. This is what is the pharmaceutical industry that try to mimic drugs, and they try to use just generate another drug in a class that is already characterized and improve it a bit. But it's not really that it is a completely new target. And this is very, very rare. So I think glycobiochemistry and glycobiology will definitely have its its time, and it's upcoming, I think. Yeah. So, and we need the basic research for this, because otherwise nobody would ever
Understand.
like put money in this and really
Mhm.
the therapeutics are the end point, but there is so much more that has to be done before that.
Yeah. That's great.
Yep.
Yannic, thank you so much for sharing your work with us. It's really exciting, and we're going to stay tuned to see what happens next. Any other questions for Yannic before we wrap up? Any online, Rebecca? You've got one. Sorry.
There's one question from Pat G. Do scientists know how early in embryonic development the glycocalyx appears?
Good question.
Immediately. Like basically, the embryo is not developing without having the structures or the enzymes that form the glycocalyx. We know that. If they are missing, vertebrate life in general is impossible. It's embryonically lethal. Yeah.
Fascinating. Yes, one more. Kevin.
Do you know which cells produce heparanase 2?
Sorry.
Which cells
Which
produce
cells?
the heparanase 2?
So for zebrafish, we know that it's primarily cells of the liver. We don't know exactly whether it's only hepatocytes, endothelial cells, and hepatocytes, but very likely hepatocytes and endothelial cells. We also know that endothelial cells produce it. And we see it also there were studies done and it looked at expression of it, not really on a cellular level, but on an organ level. You can say that in most organs, it's expressed on a low level. Yeah.
Great. Well, thank you so much. That's very, very exciting. Switching gears just a little bit to promo our next Science Cafe, which will be on September 8th. We are going to be talking about aquaculture, so totally different subject, but some of you may recall that there have been some proposals for large finfish aquaculture here in Frenchman Bay. And so we have invited folks from the Conservation Law Foundation to come and talk to us a bit about where that process stands in terms of understanding the environmental impacts of large finfish aquaculture. And up and down the coast of Maine, Maine is actually the only state right now which has active finfish aquaculture. So we're curious to hear from them about some of the data that they have developed to take a hard look at what the environmental impacts of these large-scale operations are on our oceans. So tune in. That will be a virtual presentation. We'll have a team from Conservation Law Foundation in Boston joining us via Zoom. So that should be an interesting conversation, and we look forward to that and invite you all to join us. So thank you all so much for your attention tonight. Thank you for being here, and thank you to Yannic. Great work, and we're excited to learn more.